Today, the focus is going to be on visualization, dimensionality reduction, and manifold learning. So I know some of you have taken a look at the syllabus, and I maybe just wanted to, I looked sort of at the survey results from yesterday, and I just wanted to clarify maybe a little bit of how we're sort of approaching a lot of the things that we're doing in this course, because I think a lot of folks had questions like, what is the effect of different kinds of clustering on the way that we visualize data, or why are we using fate, or what happens if you have multiple batches? And I think all of those questions will be answered, and it's really exciting to see that people are already starting to think about the topics that we're going to be covering in the next few days. So today we're going to be talking a lot more about specifically visualization and dimensionality reduction, because this is an important way that people start to look at data. Tomorrow, we're going to talk a little bit more about some data correction strategies, either data denoising or data integration of multiple batches. And we're also going to talk a little bit about compositional analysis. If you have multiple data sets, how to identify what cells are enriched and which are depleted, those kinds of questions. Then next week, we're going to be talking about how different kinds of clustering approaches can be used to isolate different regions of the data set. And then finally, we're going to spend the last couple of days talking about neural networks. And really, I think this is an important concept to understand for really getting a sense for how all of these different kinds of sort of an important set of tools that are being developed for single cell analysis, scalable tools. But I think just understanding neural networks is very complicated and entirely different from some of the things that we're teaching so far, but in some ways very conceptually conceptually related. So I'm excited to get there. But today we're just going to talk a little bit about reducing the dimensionality of data, because one of the things that we sort of mentioned before is that oftentimes you might have data measured in many, many, many dimensions, tens of thousands of genes or dozens of proteins, but that the data lies on a lower dimensional surface. Imagine sort of like a sheet of paper in a three-dimensional space. A sheet of paper really only has two dimensions on it, but you can imagine it embedded in higher dimensions. And so one of the things we're going to talk about today is how you might reduce the dimensionality of the data set to get a representation that you can use to visualize the data, or even just as input to downstream analysis. And so to start, I want to reiterate some of the things we talked about yesterday to solidify a few of the concepts that we talked about. The first thing is that our data that we're looking at in this workshop, mostly single cell RNA sequencing data, is a high dimensional matrix. It's also true if it's ATAC-seq data. It's also true if it's CyTOF data, or if it's a large collection of patient health records. We have some sort of matrix here where you have rows representing observations. Here they're distinct cells. And columns are variables or features. Those two terms are completely interchangeable. Observations and samples, features and variables, those terms mean exactly the same thing. And we're looking at a set of genes. Maybe there's 10,000 genes that are expressed in a data set, and we want to figure out how to understand that data set. We showed yesterday that you can think about features or variables as dimensions when you're plotting things. So this is a classic two-dimensional space. If you were in high school, maybe you're familiar, you heard the phrase Cartesian coordinates, where you have x comma y. In the same way, we could say x0 comma x1, and we can have some point a0. It's a vector. The vector has two values. For x0, the value is 1. For x1, the value is 2. We can represent, you can say that A exists in this space, that E is like an element of R2, and then A0 equals this vector. This is sort of our vector representation of the matrix, or of that observation, rather. We can go beyond 2 dimensions to 3 dimensions just by adding a column to this row vector. Now, instead of just x0 and x1, or x and y, we now have x2, or you can think about it as z, and we can still sort of plot that point and see sort of the relationship for that point and other features. One of the things that we're going to play with a little bit in the coding notebooks for today is a data set I'm showing here. This is of wine cultivars. So cultivars for plants are just different kinds of, they're not necessarily different species, but they're different varieties. And so you can imagine like Pinot Grigio versus Pinot Noir are two different kinds of grapes. And so those are different cultivars. And this data set is a classic machine learning data set collected by the University, I think it was University of Illinois, actually. I need to check that. It's a classic machine learning data set, and there's about 13 different features across 100 or so different wines. And here we're showing 2 of them. We're showing alcohol and hue, and each point here is a different wine, and we're showing the value for each of these 2 features. And the reason that I'm showing you this is that I think it's a lot easier to understand what features are or what variables are or how you should start to think about them when you're thinking about a lot of these physical properties of liquids, right? At the end of the day, these are liquids that people have measured alcohol content, have measured the hue of. These are quantifiable things. And just as we said, our data is high dimensional in terms of genes and cells, this data set's also somewhat high dimensional in terms of features and wines that have been sampled. I want to point out that there's a lot of different kinds of structure that can exist in a data set. So here we have basically, if you didn't see the colors, this is kind of like one big blob. But in 2 dimensions, it's possible to have many different kinds of structure. For example, you could see here a very strong negative correlation between alcohol and hue. This is a valid combination of patterns in 2 dimensions. And think about all the different kinds of sort of correlation plots you could imagine between variables. There's many, many different ways you can see that. But one of the things that's really important to understand, which is that as you have more features, you get more structure. There's more axes along which the data can vary. And so as you increase the dimensionality of the data set, you increase the structures that are possible and potentially also increase your resolution. For example, what we see here is that cultivar 2, if we look at the y-axis, the x and y-axis here are the same as that 2-dimensional plot, but now we've added a 3rd axis of color intensity. And that cultivar 2 actually has much higher color intensity than cultivar 1. And so actually, by adding this feature, by considering this feature, it might now be easier to discriminate between those 2 cultivars of wine. It's the same thing for single cell sequencing data. The reason we care about more dimensions, more genes, is that if you have more genes, it may be easier to better distinguish between populations of cells. And we've had this problem in the lab. You do a CyTOF panel for T cell markers, but then you find some cells in there that you're interested in actually look like they're B cells, but you don't have those protein measurements. You can't really distinguish what kinds of cells they are because you don't have those features. But there's a problem that happens as the number of dimensions gets incredibly large. If you have only 3, it's really easy to create a scatter plot and identify that structure in the data that, say, separates cultivar 1 from cultivar 2 here. But if you have 20,000 dimensions, you can't look at all sets of, even if we imagine you had a triaxial plot, so three features, and then your color is a 4th dimension, still you cannot look at all the possible, even just pairwise combinations of features, practically speaking. And so we need a principled way to sort through the data and identify structure without necessarily, and so that exists within the data set without having to do this manually. And this general task of identifying sort of lower dimensional representations of the data, simplifying the data from 20,000 dimensions into either 2 or 3 that you can plot on a scatter plot, or maybe 100 dimensions that you use as input to build your graph from. That task of going from many to fewer dimensions is called dimensionality reduction. If you'll remember from the unsupervised learning section of the first lecture, we mentioned that the goal of unsupervised learning is to learn an embedding of data X to some domain Y, where the dimensions of X is much greater than the dimensionality of Y. So it's natural to think about some kinds of unsupervised learning as a dimensionality reduction task, although maybe you don't necessarily want to visualize clusters on a scatter plot, but it's still reducing the dimensionality representation of the data. And so to get a sense, to teach you guys sort of how we think about reducing
the doctor's appointment to assess.
Sorry, just everyone make sure you mute yourselves. To get yourself a little bit acquainted with the idea of dimensionality reduction, I'm looking again at some cartoon data here measured in 2 dimensions, feature 1 and feature 2. And I'm going to imagine now a 1-dimensional line. A line is 1 dimension. There's only 1 axis here it can vary on. And I want to talk about reducing this data onto that line. In machine learning or in statistics, the language that we use to describe that is called projection. We project the data onto a line. And the way that that works, imagine, we're just rotating the data from here to here. And now we have a set of points that, the same distribution relationship between the points, but we're now just tilting the view. And I want you to notice one thing here, which is that this in the two-dimensional representation of the data, this red point and this green point are actually the furthest from each other in the data set. Now, what happens when we project the data onto the line is we only take, we take this and we find the closest point on the line, and that's where the point goes. Actually, it's like an orthogonal projection, but imagine just the closest point on the line. And then you get this. So this is the data. We've gone from 2 dimensions, projected it down to 1 dimension. But the problem, as you can see, with projecting this data set onto this line is that the 2 points that were the most farthest apart from each other now are immediately on top of each other. So structure that existed in this data set in 2 dimensions has been lost by projecting it into 1 dimension. Now, that's always going to happen by necessity. By doing dimensionality reduction from an information theoretic standpoint, you have to lose some information, assuming the data is not, there are no just sort of extra features that have no values, right? If this data was naturally on a line in 2 dimensions, you might not do that, but generally speaking, you lose information. And so you might say that, I actually care that these points were farthest from each other, that this is where most of the variation in the data set is. I'd like to preserve that. I would like to keep red and green far away from each other. They were the furthest in the original data set. And so I'm going to start right now with a quiz. And I've shown you one line. We just looked at B, but I'm now adding in 4 more. And I'm saying that if we could project this data set onto one of these 4 lines, which do you think is the best line that we could project the data onto to preserve that kind of structure that would, for example, keep the red and green line separate from each other? I'm going to wait until we have about 50 responses here. We've got 140 people on the call, so I'm sure we can make it. So please remember to continue asking questions in Slack. I think that's been a super helpful way to sort of keep track of what people are interested in. And I hope that that's been a helpful thing for everybody here. I've seen a lot of activity on Slack. And so, thank you everyone for asking questions. And thank you also to the TAs who've been answering them. Okay, great. So the overwhelming response here is D. And I agree with you. I think that D is the best line that we could project the data onto. I would like to emphasize that best is a subjective term. It's a subjective description. And so, I may think that that's the best, but there are other valid reasons to consider other lines here, maybe the best line. But for the way I was describing this of keeping far apart points far away, D is the correct answer here. So if we take D and then we project the points onto it again, what you'll find is that now the axis of maximum variation in the data, so if you were to calculate the variance of this distribution of these points here, this is the largest possible variance that you could get. So most of the variance of the data is explained by this line. When I say variance, I'm talking about the statistical meaning. So some of you may be a little bit familiar with the concept of principal components analysis, or you've heard this phrase before. It's very common to visualize maybe flow cytometry data in terms of their principal components. And this is, I think, probably one of the most common dimensionality reduction tools out there. And the idea behind principal components analysis is a dimensionality reduction approach that identifies axes of variation within the data set such that each successive axis is, one, orthogonal to all other axes. So orthogonal just means forms a 90-degree angle. If we were to calculate the angle between PC1 and PC2 here, that would be orthogonal. You can imagine if there was a third principal component, it would be coming out of the screen at you, and it would also form a right angle to PC1 and PC2. this is a concept that generalizes to 10,000 dimensions just as easily as from 2 to 3. And PC1 explains the most variance, and PC2 explains the next most variance and is orthogonal to PC1. So PC2, you can imagine, also could freely rotate in that axis and explain different amounts of variation, but this one explains the most. And so there's as many principal components as there are features in the data set. Often we only talk about taking the first, 10 or 20 or 100 principal components, or 2 if we want to do visualization, but technically it's a full decomposition. So you can actually get as many principal components as there are axes of the matrix. But now sort of there's this question, right? In 2 dimensions, it's relatively easy for me to pick that line. But if I just have this data set, how would I identify what is that axis of maximum variation in the data set? And in order to understand that, one of the concepts that you need to understand is this idea of covariance. So variance of a variable is just the expectation of the difference between the observations and the expectation of the variable. If you haven't taken a class in probability theory, you may not be familiar with this phrase, the expected value of a random variable. That's because it's equivalent to the concept of the mean, or the arithmetic mean, or the average value in a data set. You could consider as the expectation of that data set. A random variable is not a set of observations, but rather a distribution over a domain. And so you can take that, sort of center of mass of that distribution, that's the expected value. And then depending on how far away those points are, the sum of the differences, or the average distance from that center of mass, is your variance. And when you're looking at covariance, this is generalizing that to a 2-variable regime. So we're looking at the covariance between 2 random variables, xi and xj. And what we have here is the expectation of the product of the variances in each of the data sets. So it's not exactly the variance, but the difference between xi and the center of mass of xi, and the set of differences between xj and the center of mass of xj. And to sort of get an intuitive sense of this, you can calculate the covariance between every pair of variables in a data set. And covariance is high when the 2 variables are varying together. So when increases in one lead to increases in another, sort of an idea of correlation is sort of, they're not the same thing, but there's some similarities in these ideas of like strongly associated covarying variables. The more associated they are, the higher the covariance should be.
I would just quickly summarize by saying the variance is how much a variable varies from its mean. If all the values are centered around the mean, the variable has little variance. If all the values of the variable are scattered on either side of the mean really far, it has a lot of variance. And this matrix is computing either the variance along the diagonal, or what's called covariance, or how 2 variables vary from the mean together in the matrix.
Yeah, exactly. And I think if we think back to the kernel plots, we were talking about kernel bandwidth. Bandwidth and variance are very tightly related. So if the bandwidth is very wide, the variance is very large. So I think these are all very interrelated concepts. Thank you for clarifying that, Smitha. So covariance is one thing. So we have a matrix now that for every pair of variables, so this is x1, x2, this is x1, xn, here we have xn, x2, every set of pair of variables, we can look at how much they vary together. And so I want you to maybe hold on to that idea for a second. I need to introduce another concept first before we get to how PCA works, which is the idea of a matrix as a transformation. So we've talked before about a matrix as representing a data set where the rows are observations and the columns are features, but a matrix is just a generic container for numbers. It's a structured representation of numbers, and it can have many different interpretations. So one of the most important representations or functions of a matrix is a transformation. So you can imagine this as shifting some of the features in a matrix. And one of the easiest ways to sort of think about this is a rotation matrix. So a rotation matrix is a special kind of matrix that has cosines on the diagonal, on one of the diagonals, and then so this is in 2 dimensions. It's different if you have more, and then you have sine and negative sine on the off diagonals. And this value in the middle here is the amount of rotation you want to do. So if you have a rotation matrix and you represent it by a line, this line might have the value 1, 1. So right, you can represent a line or the point as a vector. If you multiply this rotation matrix by the vector representation of this line, what it does is it rotates that line 90 degrees. And so this is a rotation, this is a matrix that is an operation on a variable or on a point. So you multiply a point by the rotation matrix, it rotates your point. This is actually exactly how, if you ever use something like Photoshop or Illustrator and you say apply a transformation to rotate or scale a point, right? If you make it wider or taller, these are all transformations. Those are applied using matrices that look just like this. Next slide. Great. And I'm just sort of highlighting, what I was saying before. This point could be represented as 1, 1. This rotation matrix times this vector produces this vector, negative 1, 1. So this is our 90-degree rotation. So you take the matrix product of this matrix and this vector, and you'll get these 2 values out. And this produces, I'm realizing there were animations here that I forgot about, but these are vector notations, and this produces a line like we were seeing before. So x1 would be negative 1, so it'd be out to the left here, but x2, or the y-axis, is still 1, so we only get sort of a 90-degree rotation counterclockwise. So I'm talking about the idea of a matrix as a transformation. So a rotation matrix is a matrix that only rotates lines in one direction. So you take any line and it will always rotate it 90 degrees or 45 degrees or 30 degrees. And any line will just get equally shifted. But there are other kinds of matrices that don't have this special structure here with cosines and sines, that will apply different transformations depending on where you are. So this is a vector field that says for each point how far away that point is going to get pushed by a particular transformation matrix. There is a property of a transformation matrix that there are some vectors that when you apply the transformation to them do not get their directionality does not change, only the distance, they only get scaled linearly. Their distance from the origin changes. And so if you look in this plot, if you notice along this diagonal here, there are arrows that just go straight out. So they're this, if you're imagining a point here at 1, 1, the result of applying the transformation represented by this vector field would just be to push it out in the same direction. Contrast that with, for example, this point here, the transformation matrix is going to push it out and up. So that's a little bit different than what we're seeing here. I know some of these ideas are actually
vectors don't rotate the line, basically, just using rotation.
Yeah, so for using the rotation context, yeah, you can imagine that most transformation matrices will include some kind of rotation for almost all of the lines, with almost all points as input, with the expectation of the eigenvectors of that matrix, which only get stretched. There is a really great series of videos from 3 Blue, 1 Brown, or 3 Brown, 1 Blue, I can't remember exactly what it was. It's in the workshop materials on eigenvectors. I highly recommend it if this sounds at all confusing. Eigenvectors are very confusing. This is usually the last thing taught in a semester's long college course on linear algebra. So if this doesn't make a ton of sense right now, please understand that's okay. And we can send, yeah, it is 3 Blue, 1 Brown. Shari just posted it in the chat. Highly recommend, really great. And also actually has a really good concept on eigendecomposition and PCA in that same series. So there are these special vectors in a matrix that if you apply the matrix to those vectors, they do not rotate, they only get stretched. Their magnitude changes, not their direction. So a square matrix can be decomposed into a set of these kinds of vectors. So there's the vectors that don't change and a set of stretch. So the vectors are called eigenvectors and the amount of stretch is called the eigenvalue. So you can decompose a matrix into a set of eigenvectors and eigenvalues such that when you multiply all of the eigenvectors by their eigenvalues, you can reconstruct the original matrix. And so, yeah, I'm seeing some other people in the chat are recommending that course. 100% say you should do this. Eigendecomposition works for, so this is for the same idea, right? So we have a matrix, we can decompose it into a set of eigenvectors and eigenvalues. And if or sorry, I guess this is actually SVD, isn't this?
The same idea. You can take a matrix and that's a transformation matrix, and you can reword it as a matrix that rotates to the eigendirections, just stretches and rotates back. That'll be the same transformation as just transforming, applying that transformation to a vector.
Exactly. So you can take, so as Smita said, we have now one matrix and we broke it down into a set of vectors and amounts of stretch that can be used to rotate, push, and then rotate back the data into the original matrix. And the question is, okay, great. So I say you can do this, you can identify these vectors. You know, we were just talking about covariance before, like why are we now talking about these vectors that don't change in their transformation? And I get that it's not the most straightforward, but what I want to point out is that there's a really important relationship between these concepts, which is that if your matrix is a covariance matrix of a data set, and we showed you before how to calculate covariance between all of the variables in a data set, and this is a square matrix, right? Because you have m features on the columns and m features on the rows. You take the eigenvectors or the eigendecomposition of that covariance matrix, and if you sort the eigenvectors by the eigenvalues, there's a property that emerges where the eigenvectors are linear axes with linear combinations of the input features that explain the maximum variation in the data set. And it's a little bit confusing of how exactly that proof works. But the way that I like to think about it is that the covariance matrix says that these are, if there are axes that are really tightly related and have a lot of spread, their covariance is quite large. And so if you find that matrix representation of those variables, the eigenvalue is also going to be quite large. And so you can find these axes of variation within the data set.
So Anna, can you go back to that slide? So one way that we're looking at it here is that the covariance matrix is a matrix that is a transformation, and it takes a circular blob and it turns it into data that's shaped like your data based on the covariance. So it creates the covariance in your data. So if you look at this matrix, in some directions, the data has to be rotated to fit the shape on the right, and in some directions, it just has to be stretched. And it turns out the direction in which your data is stretched the most is the largest eigenvector. And that actually also represents the highest covariance. So if you want a matrix that takes any old random data and turns it into your data, that matrix is going to have to stretch where your data stretches the most. And that's the largest eigenvector.
Yeah, yes. I think that's another good way to think about it. So this is sort of a random data set, and this is the data that we want. And so this is you'd have to take this data and stretch it to get it to be this axis. I'm sorry, actually I'm realizing that's not the right mouse, this axis. Okay, so one of the things we were saying before is that these vectors, the principal components that we want through the data set, are eigenvectors of that covariance matrix, where the first principal component has the highest associated eigenvalue or the greatest stretch associated with it. And then PC2 is orthogonal and has the second highest eigenvalue and has the second greatest amount of stretch. This data set only has two features, and so it only has two principal components. And so as you can see, there is less stretch in this in PC2 than in PC1. So this sort of explains how we're going to get that representation of the data. So I'm going to show you guys now a new data set, and I would like you to predict which do you think is going to be PC1 for this data set. So this is not the same data set as before. A, B, C, or D? Great, so we got about 50 responses, perfect, awesome. So almost everyone said D. So yes, PC1 would be D, and PC2 would be B. So this is the second greatest axis of variation. So this is, basically the whole point here is just to sort of cover this basic idea of, if you wanted to have a data set, you identify the most axis of variation. If you had to reduce from 2 to 1 dimensions, you'd want to use PC1 or line D here. But now, what if I gave you a different data set that looked like this, right? We said there's many possible data structures that can exist in 2 dimensions. So here I'm showing you a circle, and maybe the circle is not a perfect circle. But what if I wanted to project this data into 1 dimension? How might I do that, right? And so if it were a perfect circle, it would be, basically any possible linear combination of x and y would be our eigenvector, our PC1.
Equally bad projection.
Yeah, exactly. Be equally bad, right? But like really, you can imagine we're talking about just dimensionality reduced representations. And so you don't just have to do a linear projection onto a line to get a dimensionality reduced representation of the data. There are other nonlinear ways that you can do this. Although I will, they're slight with increasing levels of complexity. Let's see. Yeah, I also just, as someone noted in the chat, it would be really great if people can ask questions in the 2021 workshop MISC help, because that way it's, honestly, even just copying and pasting links from the Zoom chat is kind of hard. And so I would just direct people to do that. I'm really glad that people are excited. That's, I think, first and foremost, the thing I want to focus on. Great, so I'm gonna hand, continue asking questions there. I'm going to hand this off to Smitha for the next section of the lecture.
Okay, yeah, if you guys have any questions, let me know on the previous section. So the previous section, just to summarize, is this working?
I did give you control, so maybe just try clicking on the
window. Yeah, I'm controlling the screen, but it's not actually, yep, okay. Okay, I'm gonna go back here. If you guys have any questions, let me know. Are there any outstanding questions in chat?
Yeah, we can take 30 seconds and people can just unmute themselves if you want to ask a question about this. It's definitely a complicated topic.
So what we actually covered was how to think about the idea of representing your data in lower dimensions, the idea that you will miss some information. And we also specifically covered PCA. Somebody in the chat previously had the question that, are we talking about the PCA plot? The PCA plot is actually just the projection of data into those PC components and then using those PC components as the axes. So it kind of rotates your data into those PC components. You're looking
I have a question.
Just let me just finish that quick thought. If you're looking at a PCA plot, PC1 will definitely have the most variation. But go ahead. What's your question?
Yeah, I'm a little rusty on my linear algebra, but like, weren't there certain matrices that you couldn't decompose with the eigenvalues and vectors? If that's the case, what do you do?
So there are rectangular matrices, and they have a similar concept called singular value decomposition. But they're not as precise as eigendecomposition. But the covariance matrix has dimensions features by features, so it's a square matrix, and so you can eigendecompose it. You can eigendecompose any square matrix for the most part.
Thanks.
And the concept of eigenvectors actually turns out to be important in the rest of the lecture as well. So the way we described eigenvectors, just to reiterate it again, is it's really a concept that's about linear transformations. Matrices, like the covariance matrix, can be a linear transformation. Your data matrix is not a linear transformation. The covariance matrix actually can be thought of as a linear transformation that transforms some data set to have the covariance of your data set. The eigenvectors are really sort of the independent components of linear transformations, where linear transformations can be thought of in terms of what are their eigenvectors, or what are the directions that they just stretch. That turns out to determine a lot about those linear transformations. And eigenvectors are also orthogonal from each other, and they form an orthogonal basis for your data. And in that basis, the transform just stretches. Somebody else has a chat. For some reason, I can't see the chat in my Zoom right now. Can you read that out to me, Dan?
Yeah, it just says, what about covariance matrix over sparse data?
So the covariance matrix has dimensions features by features. So I'm not sure the question. So if your data has 10 features or something, 10 by 10,
Maybe,
you could have.
If you ask that question on the Slack math help, we can maybe help there. I think there might be a little bit of a misconception, but the sparsity of the data set does not have any bearing on your ability to calculate covariance. You can calculate it between any set of variables, regardless of how sparse
They
they
might be talking about missing values or missing features. So usually you won't have things that are not numbers when you compute these matrices. So you'd have to fill them in some way, or interpolate them in some way. We'll be talking a little bit about denoising and filling in values in the next lecture. Or you can take out that data point for the purposes of PCA computation. Okay, so next I'll be talking about nonlinear dimensionality reduction, which will allow you to sort of find the nonlinear coiled dimensions in the data set and project your data to, for example, maybe a circular dimension that goes like this. That's maybe better able to uncoil the data and explain the shape and the trends in the data a little bit better. Nonlinear dimensionality reduction often involves learning to various degrees of accuracy, something that can abstractly be called the data manifold, or the shape and the space in which the data naturally lies. When I'm talking about a manifold, we're really talking about this idea that you want to capture as much of the structure in the data as possible. But you do have an assumption that the data lies in a lower dimensional subspace. So it's a little bit like a sheet or the surface of a balloon or something that lives in 3 dimensions, but it's really a 2-dimensional object that's sort of coiled. And the idea of manifold learning is that usually when you have this kind of smooth surface, locally speaking, Euclidean distances and some related distances do work. So you can find your neighbors using Euclidean distances, but then in order to find the whole shape, you kind of have to follow along by going from neighbor to neighbor to neighbor. And this is exactly the idea that we started to talk about mathematically. How do you accomplish this? Is generally by re-representing your data as an affinity graph or a nearest neighbor graph. And then there's a variety of ways to enhance that. For example, using diffusion, which we'll talk about. And the concept of eigenvectors will come back when you're actually thinking about, what is the dimension to project onto. But first we'll talk a little bit about the re-representation of the data. Just a little note here. When we talk about data manifolds, this idea comes from Riemannian geometry. So there is this idea of locally Euclidean subspaces or smooth subspaces that are coiled or curved in space. So what does this have anything to do with the data we're representing? It turns out that in particular biological data, but a lot of naturalistic system data has this kind of nonlinear coiled structure. And the reason is because you have some kind of a continuum or continuous process that's generating the data. If you take differentiation or response to a drug, for instance, you'll have different cells you measure that'll be at different stages of differentiation. And so from that, you'll have neighbors who are in slightly different stages. And then if you follow along the neighbors, you can get the shape of the whole data. And so you'll often see cells in, extremal states as well as intermediate states. And that gives you a good way of learning these. And in fact, these turn out to be good representations for processes that actually have this kind of continuous structure. And most biological data does. Your cells, almost none of them are identical to each other. They all vary slightly, and they take up a good chunk of the state space, usually, when you come up with a single cell experiment. So a data graph, which again creates sort of a mesh like this, will learn the underlying shape of the data. I hope that visual intuition is going to help you there. And so biological data sets have smooth transitions between states, which in single cell data is translated to small changes in gene expression. This will allow you to find neighbors, because you can find neighbors using something just like Euclidean distance. But then the larger shape of the data, we'll talk about how you get that just by being able to identify the neighbors. The basic idea is just with neighbors, you can create a mesh like this, and you can kind of walk through the mesh to get the larger shape of the data. So just a little bit of review of the concepts that we talked about yesterday when we talked about the second representation of the data, which is not in tabular form, but as an affinity graph. So the steps that we undertook were, we had our data and we computed a distance matrix. Remember in manifold learning, for example, Euclidean distance. Euclidean distance is only really a valid distance in these coiled dimensions at the very local scale. So what we did was we took the distances and passed them through this kind of kernel that inverts the distance so that small distance becomes high affinity, and then it quickly drops off. So you get a matrix like this called an affinity matrix. So this is how you get sort of a soft version of a nearest neighbors graph with weighting on each of the neighbors. And the weighting is given by the affinity. And in order to go from the distances to the affinities, we made use of these types of kernel functions. And kernel functions have this property that they're high in the middle, they integrate to 1, so they drop off quickly. Okay, one thing that I mentioned yesterday was this idea that you have the bandwidth parameter, or the sigma from the Gaussian distribution, and this will really determine how much connectivity is produced and what is the range of distances that you're going to connect in your initial affinity graph. Okay, and so this question is asking, which value of sigma produces a graph with the least global connectivity? And that's right. So the answer A would be right, because in answer A, when the bandwidth is very small like this, you have every point only connected to itself, so you're getting a disconnected graph. You start to get global graph connectivity pretty quickly, for example, in this 1.0 sigma setting. If you increase your bandwidth too much, you're no longer able to learn the shape of the data because you get these spurious connections that don't follow the data manifold. And that's what this question is really intended to say, that you actually have to play with the idea. And so at the end of the day, you have an affinity matrix that's determined by the kernel and all the bandwidth parameters that you're choosing. And changing the kernel width will change this matrix. So this is a parameter you have to play around with. And this is the case with most nonlinear dimensionality reduction methods, is that they do have more parameters that you have to set. And this is part of the model that you're learning of the data, and selecting the model is sometimes more of an art than a science, but it's still an important step to do to get meaningful results from your data. So now, how does very basic nonlinear dimensionality reduction works? It works very similar to PCA, except for you're going to take the eigenvectors of your affinity matrix rather than your feature set. So if you take the eigenvectors of your affinity matrix, those will be nonlinear dimensions that follow the shape of your data. this will become a little bit more clear when I talk about diffusions. But the idea here is if you want to go along a path where the matrix is basically pushing you along the path, it's pushing you towards near neighbors, then the path in which you just stay along it and extend yourself further on the path will be the first eigenvector. Kernel Pca is sort of a nonlinear extension of Pca, where instead of using the covariance matrix, you use an affinity matrix. The very tricky thing is kernel Pca is actually a nonlinear extension of something called multidimensional scaling, where you're looking at inner products between points instead of features. So it's like turning the matrix to the side, where you have observations by observations and looking at the eigenvectors of that. But nevertheless, it's called kernel Pca, and the idea is the same. So you take, instead of taking covariance matrices, you take affinity matrices and you use their eigendimensions as your new kernel Pca dimensions. This is the basic idea behind a lot of nonlinear dimensionality reduction methods, several of them. And we'll talk about two specific variants, which are diffusion maps and Laplacian eigenmaps, which turn out to be very useful, for example, for clustering the data or for understanding progressions in the data. But they're not necessarily specifically optimized for visualization. So the first method I'll talk about is diffusion maps. So diffusion maps do something a little bit beyond just computing the affinity matrix. So in computing a diffusion map, you go from the data. Here, the data is in 2 dimensions. You compute the distance matrix, which is pairwise distances between all points. Now you turn the distance into an affinity, so you start to get affinities that drop off quickly. So your strong connections kind of go along the shape of the data. And you could already eigendecompose this matrix and get some kind of kernel Pca. The diffusion maps go a little bit beyond this in order to denoise and really coalesce to the main nonlinear dimensions of the data. And the way they do this is on top of this graph that you've defined, they define another probabilistic affinity matrix or probabilistic transition matrix, which is a Markov matrix. And the way they do that is they take your affinities in each row of the affinity matrix, you have some affinities to all the other points in the data. What they do is they just divide by the sum of those affinities, and then you get probabilities. You get a probability that something is a neighbor. And it'll be a very high probability if your affinity was high, and a low probability if your affinity was low, because it's just a normalization on the affinity that turns them into probabilities. Now, the next step the diffusion maps use is they actually use a random walk based on these probabilities. So you're standing on a particular point, and you have a particular probability of jumping to this neighbor and a particular probability of jumping to this neighbor. A random walk on a graph is also called diffusion on a graph. And it turns out if you had an infinitely number of points, it would be equivalent to heat diffusion, and that's why it's called diffusion rather than random walking sometimes, but they're exactly the same thing. So if I was at this point, we have some probability of jumping to this point, point 4, and that means that this point in the feature dimensional space was actually more similar to this point. So you're jumping to similar points with higher probability, jumping to dissimilar points with lower probability. It turns out this kind of a Markov transition matrix that you get from diffusion is very useful because you can sort of clean up your affinities and really hone in on the nonlinear coiled dimension of the data when you eigendecompose it as well. So there's still basically some kind of nonlinear affinity computation followed by eigendecomposition, but it includes this random walk diffusion step, which I'm going to explain what that really does in a second. And it turns out this is very useful for single cell data because single cell data in particular is so noisy. So let's take this 2D matrix. So this in single cell sequencing terms would be cells by cells. So each dot is a cell. Here's a cell by cell distance matrix. We talked about the idea that you see these bands in the distance matrix because opposite bands are artificially close here in Euclidean distance. But within the manifold, they're quite far away, but we haven't learned where the manifold is yet. We haven't learned that this is the shape of the manifold and that the blue is actually quite far from the green. We're just measuring Euclidean distances. When you go to an affinity matrix, these bands lighten up because the localized affinity is really emphasizing here. But depending on how you set the bandwidth, you might not know the exact right bandwidth to set, or the same bandwidth might not work in all parts of your data. You'll still get some of these cross-cutting connections that are active. So this connectivity or this affinity matrix still has some spurious connections in it that you don't necessarily want if you really want to understand this coiled dimension here. So instead, what diffusion maps do is they normalize this affinity matrix. So these are all probabilities. So you divide every row by the row sum, you get probabilities. And this probabilistic Markov matrix is powered to some power t. And what happens when you power it is you get probabilities of t-step random walks. So instead of just getting the probability from going from here to here in one step, if you power that matrix, you'll get the probability of going from here to here in 2 steps, 3 steps, 4 steps, 5 steps, whatever it is that you power it to. The higher you power it, the cleaner your connectivity becomes. The reason is there is some probability of going from here to here, let's call it 0.1. But if you're taking 2 steps, the more likely 2 steps you're taking are going to be overwhelmingly within here. So there are so many 2-step combinations within here than any 1 step that goes in this way. So eventually, the probabilities along the density of the data are going to dominate so much that because of the normalization, these cross connections will basically disappear, if that makes sense. If you're taking n steps, it's so much easier, so many more combinations of n steps that you're taking here than any step that goes through here, because there's just one edge here and there's like a million here. So the longer your walk, the cleaner this connectivity becomes. And so at the end of the day, just by powering this Markov matrix, you've computed t-step diffusions or t-step random walk probabilities between every pair of points. And the pairs of points that occur within, the paths that occur within the density of the data will outnumber paths that go through sparse areas by so much that your probabilistic affinity is going to become very dominantly in the shape of the manifold. And that's really what diffusion does. So once you have your diffusion powered Markov matrix, so if you power your matrix, how do you achieve a t-step random walk? I'll describe it in a second. Probably the next couple of figures will help you see that. So the basic idea is if you're going from 0.1 in 1 step, you're going to end up in 0.2, 3, 4, 5 based on the probabilities described in the row of that matrix. Now, if you want to go 2 rows, what you have to do is you have to take 1 step somewhere and take another step. So that's basically what the matrix multiplication does. It conjoins 2 steps. It says after 1 step, if you started out in some state and you applied the matrix once, you'd go to this set of states with these probabilities. But then you apply it again and you walk again. That's basically what it is. The idea is that this powered Markov random matrix, if you apply it to a vector, it'll give you the probabilities of transitions to various other matrices. So now you've various other points. So now you've ended up at various other points. And then if you apply it again to a vector, you get the probabilities of 2 steps. And if you apply it again, you get 3 steps. So the basic idea is you're in a certain state now, and your current state can be described by a vector. I'm writing it in the chat. You apply your Markov matrix once, that mimics a 1-step walk and all the probabilities of where you'll get to. 2-step walk, 3-step walk. So, and if you just collect them, you get a t-step walk that you're going to apply to a vector. But we're not necessarily applying to any vector. We're just powering it, and that'll give you 3-step walk probabilities between any point and any other point. Does that make sense?
So how do you know how many steps do you have to apply to cover?
You don't. That's another model selection parameter, and there are some heuristics for doing that based on how many eigenvectors are significant in your Markov matrix. Because basically what happens when you power these Markov matrices, the eigenvectors stay the same. The eigenvalue changes, and the eigenvalues for some of the small eigenvalues start to disappear. As you power higher and higher eigenvalues, they start to disappear. Because such a powering doesn't change the eigenvectors, it powers the eigenvalues. And an eigenvalues of the Markov matrix all range from 1 down to 0. And because they all range from 1 to 0, the eigenvector, the trivial one that has eigenvalue 1, will stay. All the others will start to get lower and lower. And so what you're doing is you're cleaning sort of connectivities of less and less significance. And based on how much you want to sort of clean the data, you want to power it more and more. If your data has a lot of noise, you would use a high T. And if your data doesn't have very much noise, you'd use a low T. Does that make sense?
Yes, thank you. Does this become the basis for computing RNA velocity? You know, that paper by Fabian Thies. Is this
RNA velocity by Peter Karchenko's lab? The paper called that, I think, is by Peter Karchenko's lab, was published in Nature. I believe it does use a concept of a probabilistic transition, but it's computed directly from some kind of assessment of whether a particular transcript is a new transcript based on whether it's polyadenylated or not. Or if it has like splice junctions that are going to get eventually cut out or not. So that's simply looking at transcript ratios. This is the basis actually of a paper that Fabian Thies wrote called Diffusion Pseudotime, which uses the concept of a diffusion operator, which we'll talk about at some point. And that paper's basis was also in what I'm going to be talking about right now, which is called a diffusion map. And I'll get to that in just a second. Somebody asked me, is this similar to the concept of a hidden Markov model? It is a Markov model. It's not very hidden. It's a very explicit Markov model, which is describing how you would transition from one data point to another. It's kind of a Markov transition matrix that describes paths or walks through the data. If you have any other questions, please unmute yourself and ask.
Hi, what about if our graph has a cycle, then still that diffusion kernel random walk can still handle those cyclic graph?
Yes.
Okay, great.
Yeah, it doesn't matter the shape. This is completely non-parametric, other than, you know, sort of the localized affinity calculations and stuff. You can represent any shape in this way. There's no existing model. You're discovering the shape of the data by first creating a graph, then cleaning up that graph with these diffusion methods, and then eigendecomposing the resultant matrix. So this is just once again emphasizing that powering or walking more than one step on the data reveals dominant data manifold directions. So let's say you have 2 points A1 and A2 like this. So let's look at what the probabilities would be. So usually you have self probabilities, but we're looking at random walks where there's no self probabilities. This is called a non-lazy random walk, but you can look at a lazy random walk as well. So here from this point, you're pretty much going to go to this point or stay itself, but we're suppressing the staying probability, so you're going to another point. From that point, where could you go? You know, maybe these 4 points are equidistant, so you could go back, but more likely you'll go in this direction. And then if you go, let's say randomly pick this point to go to, then you could go in these directions. And eventually there will be sort of a high probability of going here, you know, maybe in a lower probability in 4 steps, a higher probability in 5 steps. So you're inducing global connectivity. But let's say that you had a spurious point here that was in between, kind of like the kind of point that would be in between the arms of a Swiss roll that's there because of noise. It's been pushed away from the manifold because of noise or dropout, which is common in single cell data. And then from the outset, you don't have a high probability of picking this path. You're going to pick paths over here. And then if you go 2 steps, you might end up sort of staying here and not completing this path, but all the paths that you have going here are going to be going through here. And so you'll get paths reflective of paths that are going through the density of the data. And that's a little bit of how the random walk matrix works, is that when you power, you're talking about taking more than 1 step. So in 1 step, you're going somewhere, and the next step, you're going somewhere else again. And it's all computed for you. All the probabilities of every place you could end up in 2 steps with every probability is computed when you just do that Markov powering. So now you have a powered Markov matrix. And now if you eigendecompose this, you'll get something called a diffusion map. And the components of the diffusion map are like your PC components, except for they're nonlinear, and they're very good at catching the coiled nonlinear dimensions of the data. So one difference between PC and this is you can't start with your first eigenvector. The first eigenvector is so-called your trivial vector, which describes on the one side steady state distributions and on the other side how many connected components you have. You can take the rest of them. The rest of them are like your PC components. So diffusion component 1, 2, 3 on, and you still have n of them. This is a form of kernel Pca, but with your connectivities highly refined by diffusion, if that makes sense. And this was developed by a colleague of ours here at Yale, Rafi Koifman, and his PhD student Lafon back in 2006. But actually, I would say it's become very popular just in the last few years, like since 2015, because it's so useful in biology. That thing that the diffusion does, really clean out probabilities, give you global connectivities and differences in connectivities based on t-step paths, turns out to really sharpen your affinity matrix and make your ability to reason about these nonlinear dimensions much better. So if you look at the first non-trivial diffusion component, you actually get a path through the data. So you can just color or label every point by its value on diffusion component 1, this phi1. If you do that, you'll see a slowly varying path where points here have low values of that eigenvector, points here have medium value, points here have high values. And this is exactly what I mean by if you just laid this out on a one-dimensional axis, now you've uncoiled the data. You've like unrolled or unwrapped it, so that now you understand how the progression within the data is, and you'd never think that blue points are close to orange points because you can see exactly how far away they are. So one of the new concepts that you get out of this is you get a new coordinate system for every point in diffusion dimensions or diffusion map dimensions. And the Euclidean distance between 2 points in these dimensions are called diffusion distances. So these are manifold intrinsic distances, and these are manifold intrinsic coordinates that you're getting. And the first non-trivial dimension is really giving you the major non-linear axis of progression. How do we know what kind of shape of manifold to expect from a data set? You don't, and that's why you have to do this computation. So you're not sure, right? You could we'll see later on that you might be able to visualize and get an idea of it. And so here's an example of a really high dimensional data set. Each of these pictures of these lips, this is actually from one of their papers, is very high dimensional. It's something like 740 pixels or something. It's like 28 by 28 images with, grayscale values. And yet this lips manifold, how many dimensions do you think that the intrinsic dimensionality of this data is? Like, what's the space that this manifold lies in? How many dimensions are there, do you think? Dami, I see your mouth moving. Yeah, something like 2. What are the 2 dimensions?
Just like the Cartesian dimensions, right?
No, the 2 dimensions here correspond to variations in the data. So there's only 2 ways in which the data is varying. The person's mouth is staying the same. Those pixels are staying the same. Yes, Arvind, you're right. So the mouth is opening or closing. That's 1 dimension. And sometimes the mouth is making sort of a different shape, like its width is changing too. It's making more like an O shape or more like a flat shape. And those are the 2 dimensions. And this is the diffusion map of it. The diffusion map shows that there's one axis like this way that's controlling open versus close. And there's one axis this way that's kind of wide mouth versus narrow mouth. And that's it. Those are the 2 dimensions that this data set lies in, and this entire data is embedded in this 2-dimensional space. So this is the idea. Your cells, too, are embedded in something that I estimate is like 20-dimensional space, even though the ambient dimensions are 20,000. And this is really what nonlinear dimensionality reduction can help you catch, is these kind of much lower dimensional intrinsic shape and the intrinsic axes of variability. What's really varying, okay? Maybe your cells are all epithelial cells. Maybe the thing that's varying is some of them are more proliferative and others are not, or some are more metastatic and some are not. So this is what nonlinear dimensionality reduction is going to help you to explore. So now I will say one thing. So the diffusion components are really fantastic at getting progressions in the data. So let's say you have this kind of data with a lot of branches. So let's assume that this is some kind of differentiation data and you're getting all these branches. What the diffusion components will do is they'll pick out particular progressions. So there's some lineage that's developing there, and it has diffusion 1 is really going to give you variation along that progression. So starting and ending of that progression. Diffusion 2 might give you the coordinates along this overall progression here, most highlighted by this branch here. But you see what diffusion components are doing, similar to PCA, is they're kind of disentangling this space. Diffusion 1 is really activated in this branch. Diffusion 2 is activating this branch, if you can see it here. Diffusion 3, this branch. So diffusion components are kind of decomposing your data into different components or branches based on the major pathways in your data. So in particular, what that means is diffusion components are really valuable, for example, for clustering. If you take diffusion dimensions and cluster, you can get clusters of all kinds of weird shapes, or for analyzing progressions or trajectories in your data, because by following along these diffusion eigenvectors, you can follow along these progressions. But they're not necessarily very good for visualization. What would happen if you projected your whole data to this? See, all the points that have 0 for that eigenvector are going to collapse on top of each other. All the points that have 1 will emerge out. So most of your points will be collapsed. And a lot of times when I've covered this class, when I haven't talked about this so explicitly, it hasn't been clear to people that diffusion maps are super valuable. They really get the intrinsic shape of the data, but they're not designed for visualization. So sometimes people say after the class, I decided to try diffusion maps instead of t-SNE and I didn't see anything. And that's because they're not really meant for visualization, because each diffusion component sort of tracks or follows a different path in the data. But they're very valuable for actually catching all the paths and looking at the shape of your data overall in a high dimensional sense. Every diffusion dimension is catching some kind of branch or progression in your data. And so that's one use of this nonlinear dimensionality reduction. And similarly with PCA, when you visualize PC1 versus PC2, there's a lot of things collapsing on top of each other. PCA also is very good. if you want to summarize your data in 10, 20 dimensions and do something on top of it, like feed it to a neural network or cluster it. Similarly, the nonlinear version of PCA, kernel PCA methods like this, especially diffusion maps, are good for further processing. If you take the diffusion dimensions and you want to analyze different progressions, different branches, different clusters in your data, they're very good at that. So I'm going to talk really quickly about another method called Laplacian Eigenmaps. They came out around the same time as diffusion maps, maybe half a year earlier. Mikhail Belkin was one of the inventors of that. He came and spoke at Yale last year. So I'm just going to talk about them, even though they're kind of similar to diffusion maps without the diffusion. They're a little bit like kernel PCA. It's because they use a slightly different matrix that helps you see something very interesting about these eigendimensions that's going to come in handy in the future lectures. Laplacian eigenmaps, instead of just using the affinity or the adjacency matrix, they use kind of an inverted version of this, where you're going to use the degree minus the adjacency. The degree matrix just is the row sum of your adjacency matrix put on the diagonal. So the row sum of this row is 1 plus 2 plus 2 plus 2 is 7, and it's just put on the diagonal, 3, 3. And so this matrix, called the Laplacian matrix, is just degree minus adjacency. And you can use your powered Markov matrix instead of the affinity matrix here too. That's called a random walk normalized Laplacian. And so if you take this Laplacian matrix and you compute the eigenvectors, you'll get something called a Laplacian eigenmap, which is very related to a diffusion map. The only difference is in diffusion maps, you weight the eigenvectors by the eigenvalues and you're powering and changing the eigenvalues themselves. In Laplacian eigenmaps, you get something really similar as diffusion maps, except for the difference is it's easier to derive a very interesting property that I'm going to tell you in a second. So you can have different kinds of Laplacian eigenmaps. You can even have one that's similar to a diffusion map. It can be the identity. If you divide both sides by degree inverse, it's going to become the identity minus your diffusion Markov matrix. So it's just kind of one off from it. So what that means is the eigenvectors of your Laplacian are actually the same as the eigenvectors of your diffusion map, particularly if you use this Laplacian. What are going to be different are the eigenvalues. They're going to be flipped. In diffusion maps, the high-valued eigenvectors were the kind of the main slow-moving components. Here, the high-valued ones are going to be the noise or the high frequency, and the low-valued one are going to be the low frequency. So why is this kind of interesting to look at? It's because in Laplacian eigenmaps, the eigenvectors give you what's called the frequency of the component. And they're used in this new field called graph signal processing because you can read out the frequency of these eigenvectors, and this will give you an idea of what kind of trend you're looking at. So let me give you an idea. And again, these can be translated almost exactly to the diffusion eigenvectors. So this holds true for the diffusion eigenvectors. But for example, the first eigenvector, which had the high eigenvalue in diffusion maps but have low eigenvalues in Laplacian eigenmaps, would be this vector. So if you take this graph, which is a line graph, like you're never going to have a line graph, but this is a fake toy line graph. The first eigenvector of the graph Laplacian of this graph has values that go from high on one end to low on the other end. This is a low frequency trend that you're describing. So if you have a gradually moving trend, like let's say these are all your sick patients and these are your healthy ones. These are your cells that are cancerous. These are the cells that are normal. Whatever it is, a trend that correlates or follows the first eigenvector is going to be a low frequency trend on your data. It's a slow moving trend that encompasses your whole data. Whereas if you look at the 64th eigenvector, it's actually jumping, jump like this. That's why it looks like an envelope. That's going to be what's called a high frequency trend. So now the noise that we often have in our single cell RNA sequencing data is the idea that phenotypically very similar cells have slightly different measurements in some axes because of noise. So what this tells us basically using the Laplacian eigenmap theory is if we take off the last few components, we will get rid of that noise. And that's actually what diffusion does. And if you want to clean your data, you can power your diffusion operator and like reapply that operator to your data. Now you clean the data of those high frequency eigenvectors that are describing noise. And this also allows you to do a lot of signal processing tasks like denoising and sharpening, which we'll get to in the next lecture. But I just wanted to mention that the frequency components that I talked about are these eigenvectors that are also prevalent in the diffusion maps, have frequency characteristics. So these eigenvectors that we talked about in our diffusion map, which are the eigenvectors of this, the first few eigenvectors are slow, long-range trend. The last few are kind of jumpy trends that change. And this is going to be very important in understanding how to denoise and smooth your data. So this is another use of kind of this nonlinear dimensionality reduction. It tells you how to do it. Here's just another example. This graph is a grid graph. This is the second lowest eigenvector in the Laplacian Eigenmap, or the second highest in the diffusion map. See, slow moving trend. This is a trend that's changing very fast. This could correspond to noise. So if whatever signal you have has high loadings onto this eigenvector, it's a very noisy signal. If whatever signal you have has a high loading to this, then it's probably a smooth, meaningful signal. Okay, so that's what I wanted to cover about the two basic kernel Pca methods. In the remaining time, I'm going to talk a little bit about methods that are specifically meant for visualization, rather than just getting nonlinear components out and then trying to cluster or do something else with them. So the reason these methods are for visualization is because they are trying to come up with the best possible dimensionality reduction that preserves as much information of whatever kind that they choose in 2 or 3 dimensions, so you can really visualize your data. And then that answers Avinash's question a little bit. What is the shape of the manifold we should expect from the data? We don't know because we can't see in 20,000 dimensions. But if we reduced it to 2 or 3 dimensions, you can start to see. So I'm going to go briefly over these 3 methods, T-SNE, UMAP, and FIT. T-SNE stands for T-distribution stochastic neighbor embedding. The goal of T-SNE, so of course, there is no possible way you're going to preserve all the information about your data in 2 dimensions if you had 20,000 to begin with. There's just no possible way. So you have to pick and choose what you're going to preserve. What T-SNE chooses to preserve based on its optimization criteria is the maintenance of local neighborhoods. So it uses a k-nearest neighbor graph. There's no diffusion or anything in there. And it's just more or less checking to see if your high dimensional neighbors match your low dimensional neighbors. So you had some neighbors in high dimensions, and you want to arrange your points in 2 dimensions so that these neighbors are for the most part preserved. And so how do they preserve it in low dimensions? Basically trial and error. They randomly lay points out in 2 dimensions, and they use gradient descent to slowly move things so that their penalty criteria is decreased, so that more near neighbors are close together. So that's why you have so many iterations when you do T-SNE. It's because doing gradient descent to rearrange it. And whenever you do something like gradient descent, same with neural networks, you get what's called a local minima. It's not globally the optimal solution you can get. It's the best solution you can get starting from that initialization. Okay, so one thing you want to be aware of. So whenever you look at a 2 or 3 dimensional visualization of your data, you want to be really aware that this is just one aspect of your data that you're looking at. You cannot possibly see all the aspects of your data. And so one thing that's a little bit concerning to me in the field of single cell is often people just use one visualization, or the most popular visualization, or the visualization that's in their favorite repository, say Seurat. But that is actually going to miss a lot of information in your data. So what I would recommend is be cognizant of that, and maybe even just visualize your data with 2 or 3 or 4 methods. There's so many methods out there to visualize. And then what's more is the earlier plot that I showed you was a fate plot. It was colored by diffusion maps. You can do that too. You can color these by diffusion component to see what's going on and what the progressions are. So at the very least, I would be very cognizant of what they're preserving. So one thing that t-SNE is not preserving is global distances. It doesn't really care if this cluster, how far it is from this cluster, because it's not trying to preserve that. There's no penalty for doing anything globally. Why isn't there? This is actually how the t-SNE penalty looks. If the probability of going from some, so it also has a Markov transition matrix, but it's not powered or anything. It has a Markov transition matrix. In the Markov transition matrix, it says, my high dimensional transition matrix says that there should be a high probability of transition. So now if I created a transition matrix out of where I've put these points in low dimensions, does it also have a high probability? So if these two are approximately equal, this will be the log of 1, this will be 0, and you won't add anything to the penalty. But notice there is actually a feature of this kind of normalization. Let's say this was already 0 to begin with. These 2 points are far away, they're close to 0 probability. Then you can place those points wherever you want. t-SNE has no penalty for it because this term's already zeroed out. Does it make sense? It's only going to care where you place this point if this was high. If this was high, you want this to be high. Okay? So somebody's asking, what are some alternative visualizations besides UMAP and FATE, and what are they good for? So there's a recent visualization, I believe, from the Bonnie Berger lab called DenseVis, and that's supposed to show some local density. You can also visualize with PCA. PCA is a good complement to something like t-SNE because PCA actually preserves global, more or less the global scaffold of data, as does FATE, but t-SNE doesn't. So if you want to see how your data fits in together besides breaking up into clusters, PCA is also okay. So those are some. One way you can think about this is in terms of a combination of attraction and repulsion. So what t-SNE is doing is it's taking points that are close together that have high transition probabilities and making them attracted to each other. But if some pairs of points have high probabilities, then Pik for some other k has to be low because these probabilities have to sum up to 1. For those, based on the initialization, it basically repels those. So what t-SNE is really doing is attracting nearby points together and kind of has a global repulsion for things that are not so near. And you can kind of see this in t-SNE embeddings if you look carefully, because most of the t-SNE embeddings you see have this quality that makes them look like they're exploding. They're like explosive because it's trying to keep some things together and it's kind of exploding the rest. And this is really the aspect of the data you'll see. You'll see what has to be near and what has to be far. And so while this is, especially when it first came out in 2008, paper by Lawrence Moderman, super innovative stuff, it still is showing something particular. It has a particular quality to what it shows. Okay, so what about UMAP? So now UMAP is actually very similar to t-SNE. So why does it have this other name? Why isn't it called t-SNE 2.0? Of course, the ultimate answer to that question will have to come from people who coded UMAP. But one thing I want to keep in mind is that it's not really published. Nobody's ever published it as a method in a peer-reviewed fashion, but when you examine kind of their preprint and you examine the method, it seems that the reason they're calling it uniform manifold approximation is to come up with sort of a topological interpretation of something that's very similar to what t-SNE is doing. So all of these methods right now are very fast. You can make t-SNE very fast, you make UMAP very fast, and FATE also very fast. So these are all very fast methods. Actually, diffusion maps you can make quite fast too, because you can do what's called landmarked diffusion, but there you have eigendecomposition. Eigendecomposition generally takes time, and none of these methods involve eigendecomposition. In particular, UMAP doesn't really learn the manifold in the way that diffusion maps or something else does. What it's actually saying is that it approximates your data as if it's uniformly sampled on some manifold. So it's creating a structure to your data where your data is uniformly sampled on that structure. So what that means is if there's a lot of points close together in a cluster that's very dense, that cluster is going to appear big in UMAP or t-SNE. And because it's kind of trying to blow out the density so that it's uniform everywhere. And that's what uniform manifold approximation means. And actually, I asked the author this, and he verified that that's what is meant by this, is that UMAP is sort of constructing a manifold that's uniform. And so the reason it does this is because they use a Markov matrix where the bandwidth is set to sort of the kth nearest neighbor. So no matter how dense the point is, it's caring about whether it keeps its k nearest neighbors close. And so you're always keeping your k nearest neighbors close, and sort of that's it. And that gives you kind of a uniform manifold feeling. Okay, and this is the density inducing variant that just came out from MIT. It's this dense map. So you see t-SNE and UMAP tend to have this uniform manifold type thing where everything is kind of blown up to be the same density, and dense map reintroduces some of the local density. And this makes it really clear that these methods don't preserve the density. So if you're doing density estimation on top of t-SNE, would want to be careful. So you really want to be careful about what the methods are showing you. Okay, so what are the differences actually from t-SNE? It turns out that they actually initialize UMAP with a Laplacian Eigenmap. So Laplacian Eigenmap is a method that learns the data manifold by eigendecomposition. So this is a good start. This is a better start than a random initialization. And somebody even showed that random initialization, somebody even showed that random initialization, instead of that, if you use Laplacian initialization, it gives you very similar results in t-SNE as UMAP. Another thing is sometimes overlooked as a detail is UMAP uses an unnormalized matrix, so it uses more the affinity matrix rather than the probability matrix. But in order to implement a penalty similar to t-SNE, it's cross entropy rather than KL divergence, it just randomly samples some things. So that means that there's random samples of repulsion and attraction. And so it doesn't have the kind of global repulsive force that t-SNE does. So UMAP doesn't look quite as explosive as t-SNE does, but otherwise it's quite similar. Yeah, I know, Dan. I just have to finish the next topic. So I'll probably be like 5 minutes over or something like that. I mean, 5 minutes from now over. Okay. So FATE is a method that we developed in our lab. And it's an alternative to these nearest neighbor preservation type visualizations because it's not doing nearest neighbor matching type visualization. So this could be a good alternative to just looking at t-SNE or UMAP, which a lot of papers look at. And after I've explained diffusion maps, this is going to be fairly easy to explain. So what FATE does is it once again computes a t-step powered Markov matrix. But now it doesn't eigendecompose it because eigendecomposition has this effect of disentangling orthogonal components or trajectories or whatever. Instead, it's trying to collect all that information into a 2D visualization. And the way it does it, and so again, you have the idea that diffusion induces global connectivity, it denoises the paths, but now you re-represent each cell by its t-step diffusion probability to other cells. And now that probability distribution that's described there is taken as a representation of this cell. So you still have the denoising, and we have a special feature here called an alpha decay kernel. The alpha decay kernel uses an adaptive density. So normal diffusion maps, you can use adaptive or fixed density. This uses an adaptive density. Because what we discovered is if you have dense regions of the data, they actually appear closer in diffusion maps if you don't do this adaptation. The reason is because if you have a lot of points somewhere, all your paths will go to that area where there's a lot of paths. So that'll actually seem like that area is closer, and it distorts the geometry. So to avoid this distortion, we use an adaptive kernel, much like t-SNE does. But don't get confused because Now the adaptive kernel is actually helping us discover the geometry of the data. You cannot interpret distance between groups of cells or clusters in TSNE and UMAP, and this is why I'm presenting another method where you might be able to interpret that a little bit more, Rohit. So Rohit's question was, how do we interpret distances between groups of cells in TSNE or UMAP since they focus on the nearest neighbors? The answer is you really cannot. And that's why I'm telling you guys to visualize with more than those two methods and use some other methods as well. So even though initially it might look like, oh, TSNE, UMAP, and FATE, they all use this Markov transition matrix. What they do with this Markov transition matrix is very different. FATE uses it as a basis for diffusion. And when you use it as a basis for diffusion, you're using it as a scaffold to figure out diffusion distances. And you don't want your diffusion distances to be dominated by dense regions, which is why it uses an adaptive density. So the reasons for using them and how it uses them are very different. So you re-represent each point by your t-step diffusion probabilities to all other points. And then FATE defines a new kind of distance between these distributions, which is a type of distance called potential distance. And the potential distance is an information theoretic distance that is actually a type of divergence between these two distributions. So if you have two cells, you have diffusion probabilities to all other cells from these two cells. What you're doing is you're taking a probability distribution distance between these two distributions, and this new distance is called a potential distance. So now we went back to a distance matrix. So we went from an original Euclidean distance matrix to affinity matrix to powered Markov matrix back to a distance matrix. And this distance matrix is the distance between these two probability distributions. And then it squeezes all that information into 2D using metric MDS. So here you see that the adaptive kernel that FATE actually uses help it discover what's called its data diffusion geometry and color it and collect it in 2 dimensions. And when we compare it on data that's artificial, you see that these visualizations all have different properties. So if you take data that's sampled along this and you add, a little bit of noise, or in this case, not much noise, and you try to embed it with these different methods, you'll get different results. FATE is first learning the long-range diffusion probability and then squeezing that in so you get something like this. PCA is projecting these to lines, so it's as if you were standing here and you projected all of this stuff to a line that's up here. So you get all of these kind of like the pink overlapping the gray and the green overlapping the red, but the global ordering is correct. Diffusion maps are really good at highlighting specific branches, so you get a very simplified looking thing where there's a lot of overlapping going on because they're separated into different components, as I showed you before. TSNE can have this shattering effect on this kind of data because it's preserving near neighbors. So these connections are correct, purple, blue, and orange, but it's kind of shattered off the global shape. Isomap is a different method that I didn't talk about. Graph layout is not really trying to preserve distances, but laying out graphs so that edges aren't cut. UMAP is similar to TSNE, maybe with less repulsion in some areas. And we see this in this embryoid body data set as well. So here you can, because of the diffusion map backend, you can really tell the trajectories that we've collected into 2 dimensions. See, PCA and FATE actually have very similar global structures. So here is an embryoid body that started at day 0 and grew up to day 27. PCA is retaining that structure. Day 0 slowly differentiates to what's at day 6 to day 9 to day 24 of this differentiation process. TSNE can sort of shatter these things so you can't follow the edges along. So for example, some parts of day 12 and day 6 are on this side of day 0 and some parts are on this side. FATE is keeping the global structure kind of like PCA, but it's denoising and clarifying the trajectories because of the diffusion process. And so we have lots of more comparisons you can look at in the FATE paper. But I want to mention that these algorithms are all basically data agnostic. A lot of people are worried that they only work on single cells or only work on this data or that data. Everything about these methods is very generic. And in fact, here's a visualization of neural network loss landscapes, like how much neural network, how well the neural network performs at each of these parameter settings as it's improving during training. So you can use these on anyone. And if you're looking to see what it is that these methods are really preserving, the best way to actually do it is to quantify it, which we're doing as part of our single cell open problems. So if your metric for your visualization is nearest neighbor embedding, then probably t-SNE or something will do better, but you can check if your new visualization that you invented is doing that. Adjusted rand index is kind of a cluster separation metric. DE-map is a metric that we came up with, which stands for denoised embedding manifold affinity preservation. And the idea of this is you take a noiseless manifold and you measure shortest path distances on it. Because in a noiseless manifold, you don't really need the diffusion to emphasize the main points. And then you add noise and you rotate it into high dimensions and you see if the embedding catches the manifold intrinsic distance. And that's something that we quantified for a whole bunch of methods. Generally, FATE was doing better. UMAP was doing better than t-SNE. Some of the other methods were doing worse, but it really sometimes depends on your data. You could quantify it with something like this. So the end of the lesson, end of this story is all these visualization methods that show you 2D visuals of multidimensional data are giving you a small aspect of your data. So, these different blind people that are feeling this elephant are all, I guess they're all blind mathematicians. One of them thinks it's a string network. One of them thinks it's a Fibonacci sequence. And so what I would encourage you to do is be really sure you understand what the method is preserving and look at different methods for your data. And somebody asked me if there are parameters to be set by the user for FATE. Yes, there are. There's the same ones as affinity matrices. So there's like the kernel bandwidth. There's also T, how many steps of diffusion. Those are the main ones. There's a couple of other minor ones. And so these are just sort of kind of a summarization that, basically PCA uses eigenvectors to identify main directions. Manifold learning identifies nonlinear patterns and structures, and every nonlinear visualization method I talked about works off of data affinity graphs. They work off of data affinity graphs in different ways. Laplacian eigenmaps and diffusion maps eigendecompose and separate components out. t-SNE, UMAP, and FATE collect them in 2 dimensions. FATE is actually built off of diffusion maps, so it tries to use the denoising and the manifold geometry discovered by diffusion maps and visualize them in 2 dimensions. UMAP and t-SNE are focused on stochastic neighbor embeddings. And so these are the main things I want you to take away from that. Any questions?
So for FATE, when it works off of the diffusion map, how many components do you use?
So we use all the components because we don't eigendecompose. We use the entire diffusion operator, but we power, right? So that diffusion operator is powered, and the powering parameter is retained from diffusion maps. It's one of the reasons why FATE is faster too, because like UMAP and t-SNE, it doesn't use eigendecomposition. Any other? How do you interpret the sizes of clusters? Does it directly correlate to the number of cells in the cluster? So on average, yeah, in t-SNE and UMAP, because they kind of blow stuff out, the size of a cluster will be equivalent to how much space it takes. In FATE, because it's trying to preserve manifold distances rather than even out the density, you could see things right on top of each other. Why do some tutorials use UMap to calculate trajectories? I can't give you a good answer to that. So I can say if your whole data set is completely connected, then preserving nearest neighbors will preserve the whole trajectory. If your data set's not naturally connected or it has points of sparsity, it'll push it out. The main reason t-SNE and UMap are used a lot just seems to be by word of mouth popularity and the fact that they got incorporated quickly into Seurat. I actually don't have any reasoning for that, given that UMap hasn't even really been published. People just tend to use what's in their favorite package, and this is what I'm trying to get you to avoid. Is powering more efficient than eigenvalue decomposition? In fact, it is, because when you power matrices, you can do what's called landmarking. That means instead of computing the probability of going from every point to every other point, you can compute the probability of going from every point to a nearby reference point, which is like a way station or railway. It's like taking the train to New Haven from New York rather than driving there. So you can take the distance from a cell to a train station to each other, which is a small matrix, and the train station back to your house in New Haven or something like that. That's called landmarking. And you can use landmarking when you're powering. Can you go back to the embryo fate graph and interpret it in addition to the... yeah, you can read that paper. There's a detailed interpretation of what this is. These are human embryonic stem cells. They've differentiated into neuronal lineages, neural crest lineages, endodermal lineages, cardiac lineages, etc. There's a whole long interpretation of it in the paper. The way you can interpret it is by coloring back by the original dimension. So if I colored this with neuronal markers, you'll see they'll all highlight up here. So that's one of the ways you can interpret it. Any other questions?
Great. Let's see. I think it might be a good idea to start breaking people into breakout rooms, and then we can, if people continue to have questions, they can stick around here. But I just want to make sure that we do give people time to get some help from TAs as they go through the coding exercises. Let's see. And so I just want to make sure. I think Scott, are you online here? Do you want to introduce the coding notebooks?
Yep, can do.
Wonderful. So I'm just going to stop sharing here, and then I think Scott, you should be able to. So Scott's going to give a very short introduction to the coding notebooks, what the goals are, and why we picked the ones for today. He's not going to go through it the same way that we did yesterday, and then we're just going to open up the breakout rooms and send you all to do your work for the next, we're going to still keep our original timeline for when the office hours are going to end. So we can just get started here quickly.
Great. So these notebooks are a little bit more split out than the last ones. It's worth noting there are 4 notebooks here, 1, 2, 3, 4, an advanced version of the last one. It's not probably any more work or time than yesterday. It's just split up a little bit more, partly because we used to intersperse in person. We intersperse the workshop notebooks with the lecture. So there are 4 parts to today's work, and I'll just show you those. First, we're just giving you a really quick intro into plotting data. So you'll be using scprep again. We've got some tools in there, and a data set called the UCI wine data set that comes from scikit-learn. It's basically some chemical properties of 100-odd bottles of wine, and you'll experiment with those features and see how you can separate different cultivars of wine, like I can't remember what they are, but they're like a Shiraz and a Pinot Noir and a Sauvignon Blanc or something like that, like different types of wine, and you can see how their chemical composition differs. From there, then we're going to guide you into creating what we're calling meta features, which means combining together multiple features to get one new better feature. So for example, if you look at the color of the wine, and you also look at the acidity of the wine. You combine those together, maybe you get something more informative in terms of separating cultivars of wine than just one or the other. And we'll use this as a stepping stone to get you into principal components analysis. So again, recommend that you have a read through the text. This is going to be somewhat repetitive of what you've heard today, but still good to repeat some of the mathematical foundations before you actually run the algorithm. And finally, after you run PCA first on the wine data, then you'll go ahead and run PCA and PSNE and FADE, and maybe UMAP as well, on the embryoid body data set that we worked on yesterday. So that's the quick overview. As always, please feel free to drop questions in the help channels on Slack. If you ask questions in your group channels, likely the only people who will see it will be your group members. And you can also always pop up to the main room, just click leave breakout and come and join us up here to ask any questions by voice.